Dear professors, I am MP Ayyappa Das, Head Department of Biotechnology from RVS College of Arts and Science, Sulur, Coimbatore, Tamil Nadu. So first of all, it's a great privilege to address all of you in this prestigious refresher course organized under PMNT scheme, which is supported by Miranda House also. And I'm thankful to the organizers, especially Dr. Malliga, for inviting me to deliver a session to the teachers. And before starting the technical session, I would like to use this platform because especially in this pandemic era, you know, the need for upgradation of knowledge in molecular biology. So the entire world is seeking and upgrading and innovating a lot of knowledge in molecular biology. So not only we, the biology teachers, even the common man, the layman, they have started using the word PCR, the word DNA, the word RNA, vaccines, covaccines, DNA vaccines, RNA vaccines, and all. So fine, as a biology teacher or as a biology scholar, we are happy that biological science is taking its high momentum, and a lot of career opportunities, a lot of new job opportunities have been created in this sector. So why I just want to share this information in this platform, because now we as a teachers, it's high time for us to refresh or to upgrade our knowledge in this stream. And I've been told that a lot of botany teachers, now itself the participation have crossed 200 in this program. So I am basically a botanist, and I have completed my BSc honors in biotechnology with a triple major system like BSc biotechnology, botany, and chemistry. So I have a little bondage with the botany subject. So I may be excused because I'm not basically a botany person, and I don't have a depth knowledge in botany when we are touching some botany part. So this is my contest, and I did my post-graduation in biotechnology, MPhil and PhD in biotechnology. And right from 2003-4, I was teaching this biotechnological aspects. And my area of research is plant DNA barcoding. So that is the general gist about my contest. So now we will try to move technically. And before starting my session, as the title of the program implies, that we know that this is a refresher course. So this is not basically a seminar or a conference, is not an orientation program. So this is a refresher course. So I just want to refresh what knowledge we have or what we are teaching from pretty long time. Or what are the things already we have in our mind. I just want you to refresh all those knowledge, and then we will try to explain what is barcoding, or we will try to know what exactly is barcoding. So before starting DNA barcoding, my humble appeal to all the teachers, because why teachers are getting refreshed or orientation all the time, the sole purpose is that this should go to the blackboard, or this should go to the classroom teaching. So basically, as a teacher, what I will tell, when whatever subject or whatever topic or whatever element you are taking in a class, so please try to refresh the previous knowledge and set a contest and go to the fundamentals and try to convince the fundamentals to the students, and then you can jump into the concept or subject, or then you can delve into the details. For example, in botany students, if there in BSc botany, if you have a basic paper called genetics, and now the teacher wants to tell about DNA, obviously in 6th standard or 8th standard itself, nowadays DNA is introduced to the students. So when you try to tell about a DNA, while you explaining itself the DNA, the entire structure or what is DNA should go into a student's mind. So first you tell the DNA, whenever you are explaining, you'll be telling it is deoxyribonucleic acid. So now the student also will started imbibing into his brain. It is deoxyribonucleic acid. Deoxyribonucleic acid. Basically, without knowing what is this ribo, what is this nucleic, or what is this acid. So once you tell if it is deoxyribonucleic acid, and now you are telling deoxy means it is devoid of oxygen. Devoid means without. So now there is an oxygen is lacking there. Devoid of oxygen is called deoxy. Likewise, each and individual abbreviations and terms and terminologies and fundamentals, if it is given to a student, automatically that will develop an interest in the student to imbibe the concept. Otherwise, the student as well as the teacher may parroting the same word like deoxy. Maybe you may be having a by heart of all these things. 2' deoxyribonucleic acid is fine. But what is this deoxy? From where this oxygen is missing? And then again you have to tell that due to the missing of this oxygen component is going to make the DNA more stable. So that again that will develop a curiosity in the students. Again the student will ask you, how sir or how madam an oxygen is missing, and you are telling DNA is the most stable biomolecule in the earth. Why it is missing, or during missing how it becomes stable? Then you can tell that okay, one oxygen is not present. So due to the lack of oxygen, the tendency or the probability for the reaction will be less. So if it is less reactive, once it is less reactive, it is more stable. So that is a universal truth. Either it is for human being or for biomolecule. If you are less reactive, you will be more stable. Likewise, if you correlate or if you make your teaching active, and if you are able to relate things with the day-to-day life, and if you are relate the concept in an interesting way or in an illustrative way, the students will listen to your lecture, and students will, apart from curiosity, more interest will be developed to the subject, as well as it will add value to the concerned teacher. So I repeat, my sole purpose of here or module 1 is not to introduce DNA to you. It is just to tell you how you can increase, how a teacher can increase his or her value through teaching, through innovative teaching or through interesting teaching. So a teacher should always gain value through effective as well as interesting teaching. Okay, that's fine. So now we will try to move to our subject. So now our subject or the topic what we have selected is DNA to barcoding. Module 1. Before DNA to barcoding, everybody or all of you are aware about barcoding. So as the slide mentions, you can see a lot of lines here, barcodes here. And if you go to any stationery store, or if you go to any super bazaar or any malls or whatever it may be, while purchasing each and every individual goods, it will hold a barcode. And you know that the guy who is there in the counter will take the object and he will be just projecting that to a sensor, and obviously that will go to the bill. So that is the new system. So that is what we call as barcoding. And even in our college libraries now we can see barcoding. And even in our identity cards, whether it is staff or students, we can see barcoding. So barcoding there, see barcoding, it is nothing but an array of codes which will lead to you specific information or what it have. It will have an array of information, and this will give you very specific information regarding that. And now the question is fine. Okay. And now how this barcoding is going to help in this molecular biology, or what exactly is DNA barcoding? So whether DNA is mentioned as barcodes, so a lot of you may have your own figment of imaginations and thoughts before arriving into the subject. So now, being a botany professors, you are all better judged than me, because DNA barcoding is all about taxonomically classifying or taxonomically compartmentalizing the plants. And taxonomical division, you know that There are a lot of taxonomy, wide taxonomy, that for systematic arrangements of the plants according to their family or according to their genera or species or whatever it may be. So the fundamental, we know that taxonomy, the science of classifying living things. So that may be plant taxonomy, or that may be microbial taxonomy, and that may be animal taxonomy. So whatever we are going to discuss hereafter is plant taxonomy. So how plants are taxonomically classified, that we know. So I think we will be able to refresh this name, Carl Linnaeus or Carol Linnaeus, the father of binomial nomenclature. The father of binomial nomenclature, where assigns each organism's a genus and species name. And we know that the rules of nomenclature, where the genus name starts with a capital letter and the species name starts with a small letter. These are all very, very, very fundamental things. I request all the teachers, either you teach in the undergraduate level or you teach in the postgraduate level, please repeat this name. Please use the word binomial nomenclature. Tell the student binomial. That means it will have two names. Bi means two, nomial means name, binomial. So again, I'm telling, then tell the student what is nomenclature. Nomenclature is a science, or it is an art of naming. Please tell the students it is the art of the naming. No matter whether he studied is in +1, +2, or 10th, or whatever it may be, no matter, you will be teaching the same thing for past 20 years or 30 years or whatever it may be. Please apprise, or please enable your learner with fundamentals. So I repeat, please enable your learner with fundamentals. Because as you know, the recent psychometric studies and psychological studies among the students, especially in this pandemic situation, the students, they quit their online class. They will be going to Wikipedia and Google simultaneously to check what is the fundamental behind. Because the teachers are always in PowerPoint presentation. They would like to discuss the concepts, and majority, they don't want to discuss the fundamentals. Having an impression the student is very clear with the fundamentals. Okay, that impression may be fine, or that impression may be true. But as a teacher, I think it's our responsibility to touch the basics each and every time, because whenever we take a survey or feedback, students will tell that, sir, please refresh it, or please recap the fundamentals, which will help us to relate with the current concept. So, and another, especially for science teachers or for botany teachers, many terms and terminologies we'll be using with the Latin word or Greek word. For example, the most commonly used word which is used in India for more than crore time per day now is vaccine. Everybody is using the word vaccine. All are running behind vaccine. I don't know in your place and all whether there is ample vaccines and all, but however, people are running from pillar to post for vaccine. Vaccine is not an English term. Vaccine is not derived from an English term. Then one student, when I was taking an immunology class in 10 years before, I was taking immunology and I was talking about vaccines. Okay, so on those periods, like 10 or 20 years before, only we biology teachers and students will speak about vaccines, maybe the nurses and doctors. But so when I was taking vaccines class, then I told the student about Edward Jenner. So Edward Jenner, the father of vaccines or father of immunization, and I told the word vaccine derived from a Greek word called vaxa. And I told vaxa means cow, because the first the vaccine experiment is performed in cow, taking the cow pustules and inoculated by Edward Jenner. So that's the history of vaccine. Then immediately one student asked me, sir, why you want to take all these words from your Greek or Latin? What is the reason? Because whenever I used to tell chemistry words or biology words, I will tell this is from Greek, this is from Latin. I've never used this is from Australia, or this is from India, or this is in our language, or this is from English. All these words are from Greek or Latin. Okay. So that was an interesting question. And again, I was also not that much aware, and I went for a hunt. And now we know that, because see, English language or majority of the language, whatever we use, in one word, there will be multiple meanings. In one word, there will be multiple meanings. But for Greek or Latin or whatever it may be, if you use one word, there will be only appropriate meaning for that particular word. So in order to avoid ambiguity, in order to introduce a new term, in order to assure accuracy, majority of the scientific term, especially biological term, it is used in Greek or Latin. So if your student, if asking the same concern to you, like all these biological terms, from where it got originated, you can tell it from Greek or Latin. And now you should tell why it is originated from Greek or Latin, which will again gain the interest of the learner. So I repeat, when you go to binomial nomenclature, we know that the scientific name of a plant or a microbe or an animal, it should have two names. And the first name is genus, and the second name is species. And I repeat, the genus name should start from caps, and the species name will start in the small letter. These are all very, very, very basic information. I know the teachers would like to listen more about that. So let me scroll to the next slide. Now the question is in everybody's mind, okay, this is known to as Carolus linearis is known to as, and classification is known to as, and what this guy is trying to tell. And initially I started with something like barcoding, and I told something about shop, I told something about building, and why it is connected to taxonomy. And we have touched the fundamentals of taxonomy also. Yes, there is a need for identifying organism, because if you want to put this entire ecosystem, the entire thing in a harmony or in a balance, first you should know what is this organism. Specifically, you should know differentiation and distinction of organism is very, very important. So there is no question in that. And I would like to put forth two concepts in front of you. One is the biological effects of global climate change and accelerating habitat destruction. So this is the two things which has been discussed and earmarked in the climate change conference, which has happened in Geneva. So biological effects of global climate change and accelerating habitat destruction. And as the slide mentions, we know only very little about biodiversity of organisms living in many unique ecosystem of Earth. Very little. But what we are telling, whatever we are seeing, we are in a mind that we are aware of all those things. Nothing. You just go to a park, you just go to a free area, just a mini forest. Each and every bird will be new to you. Each and every worm will be new to you. Each and every butterfly will be new to you. All belongs to different species at least. And now, less than 2 million of estimated plant and animal species have been identified. There are millions and millions of species, but even if we have a lot of technology hype here, less than 2 millions of the estimated plant and animal species have been identified properly. Yearly, the rate of extinction has increased, and most of them have not identified yet. So that is another very, very interesting study. That means during this era, lot of small, I think in the lifespan, lesser organisms, including plants, they live and they left this universe without leaving any trace. That means we have lost a huge data. We are not aware that these types of organisms were lived in this earth, Mother Earth, and now it has got extincted, and we are not aware of that. The huge data loss. Why there is a huge data loss? Definitely due to natural calamity, due to forest fire, due to volcano eruption, and due to deforestation, and the most threatening one, global warming. So due to all these things, we are losing a lot of plants and a lot of microbes, a lot of animals for this cause. So my topic here is that immediate and rapid mode of identification of species is required. Then I think that you have to tell, because I have put for my idea here. is there any need of revolutionary taxonomic tool? Because you will be telling me, sir, if you want to identify a plant, yes, you can identify a plant based on the leaf, based on the flower, based on the stem. That is what we call it as morphological identification. That is well happening here, and it is well done by Botanical Survey of India, and I have given a lot of samples, and they are doing it very well. And if such a system is in place, what is the need of a revolutionary taxonomic tool? Is there any revolutionary taxonomic tool? That we have to discuss. So now we will discuss whether there is any need of a revolutionary taxonomic tool. And teachers, sir, madam, now you have to tell yes or no. And based on your answer, or based on the need of taxonomic tool, our next module will start. And thank you for your patient listening. And it's me, Ayyappa Das, signing off. Thank you.